video, we shall learn about the regulation of glycogenesis and glycogenolysis. First, we shall see the glycogenesis, its regulation. How its enzyme is regulated by the hormones. So, this is glycogen synthesis process, and this will occur in the fed state. This will occur in the fed state when the blood glucose level is high. When glucose level is high in the concentration, at that time, so fed state leads to high concentration of glucose, right? So it can lead to two processes. One is glycolysis. So the level of glycolysis will increase because the cellular activities will increase, because the cell are having much amount of energy now, high ATP is now present, and it can also lead to the glycogenesis process. Okay? Now, the enzyme that is regulating this glycogenesis is glycogen synthase. This glycogen synthase has two forms. It is active form. It has active form, which we call A form. Inactive form, that is B form. Now this active form will be active when it is dephosphorylated. We know majorly all the enzymes which are phosphorylated, they are active. But there are a few enzymes which are active when they are dephosphorylated. One such is glycogen synthase. In its active form, it is dephosphorylated. And when it is phosphorylated, it becomes inactive. Okay? Now, this dephosphorylation will occur by the enzyme protein phosphatase 1, which is also called PP1, and this activation or increased level of activity is enhanced by insulin, because we know when blood glucose level is high, insulin level is also high. So insulin will cause the activation of this enzyme. So better I write it here. So plus means it is activating this enzyme phosphatase, and we know this phosphatase enzyme is involved in removal of phosphate. Like kinase, its activity is to add a phosphate group, phosphatase is to remove a phosphate group. So this protein PP1 will act on glycogen synthase B. So PP1 will act on glycogen synthase B and activate or change it to glycogen synthase A. That means it will remove the phosphate on this glycogen synthase form and activate this A. So this is how glycogenesis is regulated. Another thing is that when glycolysis is undergoing, glucose 6-phosphate is formed from glucose. When glucose is acted by the enzyme hexokinase, glucose 6-phosphate is form, and this glucose 6-phosphate also activate this protein PP1. So this glucose 6-phosphate also activate this PP1 so that this can act on this inactive form of glycogen synthesis and activate glycogen synthesis so that glycogenesis takes place, because high level of glucose is there in the blood now. So some part will go into the glycolysis pathway to provide energy to the cells, and the excess will be entering into the glycogenesis pathway to form glycogen. So this is how this glycogenesis is regulated by the hormone as well as this enzyme. Also, along with this enzyme, ATP also activate this PP1, because obviously when glucose is high, the cells are in high energy state. That means ATP's concentration is high, so it will also activate this PP1, which will in turn activate glycogen synthesis, which will form glycogen from glucose. So this is your scenario of regulation of glycogenesis. Now we will see the scenario in case of glycogenolysis. Regulation of glycogenolysis by the hormones as well as certain enzymes. So glycogenolysis is glycogen degradation, and this occurs when glucose level is low. So this occurs in, you can say, in fasting stage, when cellular energy is low. Like the cells, they have an urgent need for ATP. At that time, glycogen will be degraded to form glucose. In glycogenesis, we saw that glycogen synthesis, which was the main enzyme, has two forms: active form A, inactive form B. And the active form was dephosphorylated, and when it was phosphorylated, it became inactive. For glycogenolysis, the main enzyme was, we saw, it was glycogen phosphorylase. It also has two forms: active form, which is A, inactive form, which is B. Now this active form in this case is phosphorylated, and inactive form is dephosphorylated. You see one cycle. In case of glycogenesis, PP1 was the enzyme which was active, and that was dephosphorylating the inactive form, the inactive form to active form. In this whole scenario, that PP1 will be inactive, because in this scenario, glucagon level will be high, insulin level will be low, and insulin was activating that PP1 enzyme. So this activation is done by another set of enzyme, that is phosphorylase kinase. This also has same thing: active A form, this is active, and this is inactive B form. Active is phosphorylated, inactive is dephosphorylated. So this helps in the phosphorylation process. Now, when in fasting stage, glucagon level is high. So glucagon is high. When glucagon is high, it binds to GPCR. What is GPCR? It is G protein coupled receptor. when glucagon binds to GPCR, this activates adenylyl cyclase, okay, which in turn releases or activates cyclic AMP. It forms the secondary messenger, which is cyclic AMP. This cyclic AMP again activates, so this is all plus sign, protein kinase A. This is PKA, protein kinase A. So kinase, that means its function is to phosphorylate any enzyme. So this will now act on this inactive form of glycogen phosphorylase. This kinase has two subunits. One is the regulatory subunits, so this is the suppose catalytic subunit. One is regulatory subunit. Now this cyclic AMP will bind to this regulatory subunit, and then this will be separated. This regulatory subunit will be separated from the catalytic subunit. So as soon as this cyclic AMP is bound to the regulatory subunit, it will lead to the release of catalytic subunit. I am just drawing the catalytic form. This will now act on the inactive form of glycogen phosphorylase. So it will act on glycogen phosphorylase B, which is the inactive form. Okay? So now when this will act on this, it will phosphorylate this and activate GPA, the active form of glycogen phosphorylase, which will act on the glycogen, which will liberate glucose. So this is your overall scenario how glycogenolysis is regulated. Another thing is that when in the fasting stage, when the cell's energy is low and when it is in urgent need for ATP, at that time AMP level is also high, and AMP level also activate the glycogen phosphorylase B state. Okay, so AMP level is high. When AMP level is high, it will also activate the B form of glycogen phosphorylase. So this will act on the phosphorylase B, or you can say phosphorylase kinase B. Okay, so this will now be activated to phosphorylase kinase A. This will activate the phosphorylase active A. Okay, so this is your overall scenario. Another thing is the activation of these enzymes by the calcium ions. This activation was taking place due to the phosphorylation or dephosphorylation. Now one more thing is the activation without phosphorylation. You have muscle phosphorylase kinase. In this case, when muscles are in urgent need for ATP, so in that case, it will liberate the calcium ions. It will liberate +2 ions from sarcoplasmic reticulum. So So when a muscle is undergoing contraction or when muscles need energy, at that time it will liberate calcium ions from the sarcoplasmic reticulum, and this calcium will bind to a protein which is called calmodulin. 4 calcium ions. Note this thing. 4 calcium ions bind to one molecule of calmodulin. Okay? And forms a complex calcium-calmodulin. So this is the complex that is formed. And this complex in turn activates the phosphorylase, and this acts on kinase B, which is the inactive form. So this will act on the inactive form of the enzyme and activate it. So this is how calcium ions are helping in the activation of this enzyme without any phosphorylation. In case of liver, same thing happens, but in case of liver, what happens? In liver, during this fasting stage, epinephrine comes out. It is released from the adrenal medulla. Now this we saw in glucagon, that cell signaling by the glucagon, when during the fasting stage, when blood glucose level is low, glucagon is active and it was activating GPCR. Same here, epinephrine will also bind to GPCR, and this in turn will liberate cyclic AMP, and this will in turn release the calcium ions from ER into cytoplasm. Now same thing will happen in this case. It will bind with calmodulin and forms calmodulin complex, and this will again activate this phosphorylase kinase B. So this is your overall picture of regulation of glycogenolysis and glycogenesis by the enzymes, or you can say calcium ions, and by the hormones. So this is overall your glycogen metabolism.